Welcome children. Welcome to another topic which is very important from unit number 4 biotechnology, and that is PCR. You must have heard a lot about it, RT-PCR, reverse transcriptase polymerase chain reaction. But what actually it is? That's a very prominent question and is asked almost every alternate year. Let's have a look. Welcome to the CRISP concepts. In recombinant DNA technology, one issue that came up was how to amplify the gene of interest. If the gene is in small amount, how to amplify it? That means you need to replicate that DNA, but that replication has to be done outside the living system. That is called as in vitro replication. So a technique was devised by Sir Carey Mullis in 1986, where amplification of desired gene sequence of a DNA was done. It allows the generation of a large quantity of DNA product from only few starting copies. It has been shown that PCR can be used to generate a detectable quantity of DNA from only one starting target or a template molecule. The machine that you see here is a thermocycler, which allows change in temperature. What is the basic principle behind this? Let's have a look at it. Before that, let's see what is the application of PCR. It is used for rapid amplification of intact genes or gene fragments, generation of large amount of DNA for sequencing, generation of probes specific for uncloned genes by selective amplification of a specific segment of DNA, analysis of mutation for medical application, detection of minute amount of DNA for forensic purpose, and amplification of chromosomal region adjacent to genes of known sequences, and many more applications. Now we move on to the processes, how that PCR is carried out. In this, we find there are three prominent steps that is being followed. First thing, for replication to take place, you need to have a template, and template is provided from the parental DNA. And as we know, in case of DNA replication, the hydrogen bonds are broken by helicases, but that is no longer available in outside system. Hence, denaturation, a technique where we raise temperature to 95 degrees Celsius for 15 seconds. So what it does, the double-stranded DNA that gets denatured and it turns into single-stranded DNA that acts as template for DNA synthesis. Then comes the second step, annealing. Annealing means starting or finding a point of initiation. That is done by the help of a primer. Primer are annealed to the 3' end of each separator strand. These separator strands contains the target sequence. So the primer acts as initiator of replication. And now the most important process, the extension. So for extension, we need two things. One, the raw material, deoxyribonucleotides, as well as the enzyme that will carry it out. And the enzymes are basically protein which gets denatured at high temperature. But to wonder, one enzyme, DNA polymerase, isolated from Thermus aquaticus, that works even at high temperature. So DNA polymerase or Taq polymerase extends the primer by adding nucleotides, rather say deoxyribonucleotides, complementary to template provided to this region. And all these steps are repeated many times to obtain innumerous copies. Have a look at this schematic representation. See here, double-stranded RNA. On heating, they are converted into two single strands. Primers are being added, so that provides a point of initiation. Then DNA polymerase along with deoxynucleotides are added, which carries out the reaction. And the only thing that is differing here is change in temperature. Denaturation takes place at 95 degrees. Annealing takes place because you need to form bond, hydrogen bond, that takes place at around 62 degrees, 55 to 65 degrees in between. And extension, 72 degrees. Why? Because the optimum temperature for Thermus aquaticus is 72 degrees. So this is where it works. We can have a look at it through animation also, in vitro DNA replication. How it takes place? Denaturation. Have a look. So, annealing, joining of the primers, and then the extinction. This is repeated many times. Denaturation, separation of strand, polymerase added, primers binding to sequence, annealing to determine a starting point, and then extinction. And the credit goes to the thermostable DNA polymerase obtained from Taq polymerase, which can resist the high temperature, and it does not lose its activity even at higher temperature, and best working temperature is 72 degrees. Now let's have a look at certain questions that is being asked in the board examination. Mention the number of primers required in each cycle of polymerase chain reaction. Write the role of primers and DNA polymerase in PCR. Well, you will all answer unanimously 2, and primer starts the extension reaction. Give the characteristic feature of source organism of the DNA polymerase used in PCR. The feature is it lives in high temperature and provides a thermostable DNA polymerase. How is the amplification of gene sample of interest carried out using PCR reaction? You need to write all the 3 steps. Expand the following and mention one application of each. PCR, ELISA. For PCR, we need to write polymerase chain reaction and its application, very well aware of it. List 3 steps involved in polymerase chain reaction. Name the source organism of Taq polymerase. Explain the specific role of this enzyme in PCR. So I think now you are in a better position to answer all the questions promptly and according to the question being asked. That's all on this topic. Hope to see you again with next topic. And I am very sure now you'll not be stopping in answering the question asked in the board examination. All the best.